Had some really good talks this morning, and I'm really glad there's lots of things I thought about including, but they've all been covered so far. So how do I follow that, really? So I'm going to talk to you a bit more specifically about how you might use loss of function data to inform drug discovery. So let's just start off with a basic hypothesis. So this is sort of a kind of an idea, essentially, that if you take drug targets, you could say that a loss of function in a drug target is roughly equivalent to inhibiting or antagonizing that target. And also it might tell you a bit about the safety of inhibiting or antagonizing that target. And equally, a gain of function mutation might tell you a bit about activating a target. Of course, it may also give you extra new functions for that target. So it may also relate to off-target effects and safety and adverse drug reactions. So that's kind of what we're thinking when we've moved into this kind of research. And then if you put that in the context of validating targets, and I'm really glad about all those talks that went before, I can really skip over this. But basically, you'll often see target validation highlighted early on as something you do. You tick that box, you go on and do all the drug discovery, and that's really not accurate. Target validation really has only been achieved when the drug is safely working in the clinic for many years, and take COX-2 inhibitors and so on as examples where target validation fails very late. But genetics can actually input into this ongoing process of target validation. So the deleterious loss of function can do target ID and validation, as John was highlighting, and also as the HAO story. Deleterious mutation can also give you information on mechanism of a gene in a disease. And benign loss of function, we think, potentially can inform about safety, and talk about that more. So, as John and others highlighted, we are really moving towards the human lab. Now we're looking at studying human populations, and I would say, regrettably, funders are shutting down mouse labs around the world and around the UK. And I'd say it's maybe a little bit too early to do that, but there's certainly a lot that we can learn from humans. And so here's some examples, and you've seen other ones, of where there's a lot of papers coming out of the pharmaceutical industry saying, yeah, genetics is a great source of target validation and discovery. I don't need to labour that. And I thought about the title for this. Is genetics driving drug discovery, or is drug discovery driving genetics? I think it's a bit of both. There's a huge amount of pharma money going into genetics, like sequencing UK Biobank, Regeneron, GSK, many other companies, Genentech. They're all really very engaged in this area. So that's all great. What I'm going to talk about a lot today is about benign loss of function. And I think this is a really interesting area that maybe isn't receiving the focus that it might do. So I think a good analogy of this is the World War II aircraft. So there was a Jewish statistician, Wald, I think, who basically started logging the bullet holes in aircraft when they came back from missions over Germany. And you might think, well, there's all those bullet holes there, maybe we should put more armour where the bullet holes are. But it's where the bullet holes aren't. That's where you don't see the plane because it doesn't come back. That's where you want to put the armour. And so that's what I would say as an analogy, is that a benign loss of function mutation is like one of the bullet holes that we see. It's an area that we know we can knock out and potentially that we could modulate it for drug discovery. And where the bullet holes aren't, that's the damaging loss of function. So I think if there are damaging loss of function mutations in a drug target, then we really need to think carefully about, do we really want to modulate this target? And we need to look closely at the safety for that target. So what does benign loss of function tell us about health? David Van Heel really gave a good background to this. So the Bourne and Bradford study, this is a study that I was lucky enough to be involved in, identified lots of rare homozygous loss of function mutations in a healthy population. So it's a great resource for studying benign loss of function. And the genomes of this cohort are really interesting in that they have huge areas of autozygosity, where basically both strands of the chromosome are inherited from the same ancestor, so they have huge amounts of homozygous loss of function mutations. And David talked about this, so I don't need to go into too much detail. So there were 1,111 LOF mutations. 781 genes are disrupted by these. Now, really interestingly, 53 of those disrupt Mendelian disease genes. So there are people walking around healthy with a gene that's thought to cause a Mendelian disease. 110 of those genes are drugged. So that's the category that John was talking about. And 137 are predicted druggable. And based on John's talk, I don't even know if we can talk about druggability and druggable anymore. There's 137 of those genes are a black area, a kind of grey area where pharma might be able to do something, but I think John suggests maybe not. So what does this tell us about drug discovery? So we actually did some work for this manuscript, and one of the first things we looked at is what are the properties of these genes that have benign loss of function. And we analysed the string database, which is a database of known protein-protein interactions. And what we found, so let's just highlight a few points here. So when we look at gain-of-function mutations from disease databases, those are highly connected. On average, they have 47 connections with other genes. And when we look at the background, the standard, if you take a gene, a random gene, it will on average have 14 connections with other genes. But if you take the benign loss of function mutated genes in the Bourne and Bradford cohort, on average they have 7 interactions with other genes, so they're half as connected as average. So what this tells us is that these benign loss of function genes are much less connected. That makes sense that if you knock it out, it's less connected, so it's going to have less of a ripple effect on other genes that it's interacting with. We also took those genes and looked at their rates of success in a database called PharmaProjects, which is a database of pharmaceutical projects that are ongoing. And the bottom line here is that if we looked at the approved genes that had benign loss of function mutations, 30% of those genes got to be approved, whereas the background success rate of approval for a gene from phase 1 to market is about 14%. So again, there's this sort of doubles again. So they're half as connected in networks, and they appear to be twice as likely to go through to successfully to the market. So what we're seeing here is that genes with benign loss of function appear to be safer and surer targets to modulate. So building on the work from that paper, Dan Rhodes, who's just sitting here near the front, has been working for the last couple of years on his PhD, really looking at building and scaling up these findings and building a pipeline to analyse loss of function mutations and to exploit it for drug discovery. So he's built a repositioning pipeline that takes disease variants and looks at repurposing opportunities, looks at pathway context, but critically looks at machine learning to predict what kind of mutations exist in those genes and are they benign. So his work has mostly focused on benign mutations. So he's taken damaging and benign mutations and undetermined mutations, and then reclassified the undetermined mutations to try to identify more benign mutations. And he's using this with a machine learning approach called TPOT. So this is an overview of the pipeline. So basically, he feeds in data from a number of publications. I'll show those sources later. He's done lots of enrichments with other sources of information to predict whether or not something looks like a benign gene or whether it looks like a gene that will have a damaging loss of function. And then going through the machine learning pipeline, he's then predicted sets of benign genes that are likely to be benign in their loss of function or damaging in their loss of function. And so this is the input that he put into the pipeline. So we've got many of the papers that we've been talking about so far today. So it's about, I think a lot of the variants overlap, so I think it's about 12 to 15,000 variants. And when he actually ran the pipeline, he ends up with a classification of 1,858 benign variants, 3,600 damaging deleterious variants, and 5,000 not determined. And so when you classify these, you can see, as you might expect, that certain categories are different. So if you take the benign variants here on the left, the damaging in the middle, and the not determined on the right, you can see there are much higher rates of splice acceptor mutations in the damaging set, in the deleterious set. And the benign and the not determined set look quite similar. So it does highlight the issue that it's quite hard to differentiate the two. And it suggests to us that the not determined set might include a lot of benign mutations, but we need access to medical records to confirm that. So a machine learning approach can give us much more of a hint as to what's likely to be benign, but ultimately we need the medical record. And then also, if you look at how far drug development has gone with these different categories, these genes in different categories. So you can see that the benign and the not determined genes are less studied in drug discovery, and they're less likely to have known drugs and ligands. So as we think there might be a higher success rate in these benign targets, then it's potentially an untapped pool for drug discovery. And I think maybe another way to look at this, and thinking about John's talk, is to look very specifically at those drugged genes and to look at the difference between the drugged genes. So you can see that's the T-clin category here. So you can see still pretty low in all of the categories. So also from our initial study in the board in Bradford, we saw the lower connection of benign genes. So we wanted to investigate this in a lot more detail across this entire data set of classified benign and damaging genes. So again, we used the string database and captured all of the interactively captured the interactions around the genes. So we used network topology as part of the feeds into the machine learning. And so Dan actually built this network. So this basically is a network of all of the genes with loss of function. And you can see in red are the deleterious mutations, and they're all highly connected, as we might expect, so they tend to be more highly connected. And then the benign mutations are in green. You can see there's some interesting clusters of benign mutations here, families, which could be good for drug discovery. And then the not determined are also tend to be on the outside. So obviously this hairball here is quite difficult to actually analyse. So Dan went into some more detail, looking at different measures of pathway interaction. So he looked at things like betweenness and centrality, so how central is a gene to a pathway. He looked at closeness of centrality and degree of centrality. Now these might all sound quite similar, but they actually tell you different things about how information flows through a network. And so on this basis, here's an example looking at the betweenness of centrality. We do see differences between the benign and the deleterious mutations. So we see, as we expect, that in red, the deleterious mutations, they have a lot more of them have higher betweenness of centrality, so they're more connected. And the benign ones have more lower betweenness of centrality. And we've also then categorized these across their integration, their centrality, their closeness, and that's a log betweenness. So again, you can see all of these differ slightly. And so by feeding in the properties of what a benign gene looks like, what a deleterious gene looks like, when we've got a non-determined impact for a loss of function, a gene with a non-determined impact, we can then see how it looks in networks and feed that into machine learning. And also other Genix properties classify these benign and damaging gene sets. So for example, gene expression, again, as you might expect, the deleterious genes tend to be much higher expressed. So that's basically over the right side here, you can see that a gene that has a deleterious phenotype, when it's knocked out, tends to be much more highly expressed in a large number of tissues. And the benign tend to be lower expressed in a lower number of tissues, again, as we might expect. So having talked about humans, the human lab, I'm actually kind of surprised in looking at loss of function. I'm finding myself going right back to the beginning, really, and starting to think that we actually need to think about some of our model organisms a bit more carefully. Mouse, I've already said, I think is going to be really important, but also yeast. And the reason that I came to this conclusion is actually a few things. So this work here by Pilar in Damien Smedley's group, it shows how there's reasonably good conservation between non-essential and essential genes between yeast, human, and mouse here. And so 8% of genes that are benign in humans are essential in mice. And 41% of human deleterious genes are viable in mice. So there's some concordance, but there's differences, and we need to look at these, and we need to be aware of them. But what made me really think about yeast again, I have to say I haven't looked at a yeast paper for 15 years probably. This yeast paper made me look and made me really think about yeast again. And it's only just came out in Nature Communications, and it brings up the concept of bypass of essentiality. So basically, what they did is they showed that these genes, that if you knock them out, they're essential in yeast. If you knock it out, the yeast is not viable. But they then showed if you knock out another gene, then the yeast becomes viable again. If you overexpress another gene, then the yeast becomes viable. Or if you have a missense mutation, that might have an activating or a deactivating effect, and the yeast becomes viable. So what this is showing us is just looking at one chromosome on fission yeast. They found that 27% of the genes that were thought to be essential on that chromosome arm were actually bypassable by knocking out other genes. And so obviously, fission yeast has only a couple of thousand genes. So this is a very simple model. But then if you factor this up into mice and into humans, potentially all the genes that we think are essential could actually not be essential if you knock out another gene. It shows us biology is complex. But what I think is really exciting, and particularly for Genomics England, if you have a knocked out gene in a rare disease, you might think, well, there's not much we can do with that knocked out gene because you can't modulate it because it's knocked out. But it may well be that with models like yeast and other organisms, you could actually modulate other genes and bypass the essentiality of that knocked out gene in rare diseases. And just as you could knock out these genes with transgenics, you could also use drugs. And so you could carry these kind of results into mouse models and into humans. And in this fascinating paper, they showed the networks here. So these are the networks. The big nodes are the bypassed essential genes, and the small nodes are the genes that interact with those, that any one of those genes can rescue the phenotype. So any one of those small nodes around there, you could potentially modulate with a drug, assuming it's druggable or drugged. And taking that, the 38 essential genes that were bypassed, extending to the human orthologs, 10 of those 38 are Mendelian disease genes. 5 have drugs that act on the suppressors there. So 5 of those potentially could, in yeast at least, could have an effect on those genes. And 18 of the genes are drugged. So 18 of the small nodes are drugged. They may not interact with the Mendelian disease gene that we know about yet, but that gene may exist. And so expanding out of one of that big network you might have seen at the top. So SLM5 is the human ortholog of that, and there's a one-to-one relationship with yeast and human, is a mitochondrial tRNA synthetase, causes a Mendelian disease called combined oxidative phosphorylation deficiency, and it interacts with NARS2. This is druggable. There's a cancer inhibitor that works on that. So essentially, in yeast, you could bypass the essentiality of that and reverse a Mendelian phenotype. And what's even more exciting is that this is yeast, this is human cancer cells, and it's an emerging area. And this is a paper on bioRxiv where they've reported paralog buffering, which is essentially bypass of essentiality, I think, where they've shown that if a gene has a lot of paralogs that are close, then it's more likely to be non-essential. So their conclusion from this is that actually most genes are never or rarely essential. And I think that's a really important message to take home from this. So things like GEL and Genes in Health projects, they're great opportunities for getting lots and lots of mutation and variation data, increasing the sequencing data in healthy and diseased individuals. We need to integrate this with what we know already, OMIM, HGMD, and so on, and the disease cohorts that we're studying and population surveys. And Deepti here in the front row has done some really nice work with the Ugandan Genome Project. So here they've shown enrichment of loss of function variants in this population. And there's some examples here of variants that affect clinically relevant phenotypes like cholesterol and blood sugar. So again, more loss of function variants that could be really important for health and drug discovery. And this is the approach that Deepti is taking. We're bringing all this together in a project that we've just registered in the research environment. So here, taking together all this information, this is quite similar to what Dan's doing as well. We're taking negative training sets for function in biobank in healthy populations and positive training sets, the disease populations, and using those to inform pipelines and to better understand loss of function. And we really still don't understand loss of function very well, and current databases don't focus that well on LOF. And the LOF resource that David talked about earlier, I think, will be really important. But GEL itself, I think, needs to build a resource like this. So we're actually proposing to build an LOF catalog in the GEL environment. And this would capture things like important things like constraint measures, tolerance measures, transgenic phenotypes, gene expression, interactions, the drug discovery perspective that it's talking about, and then variant level things like ways to improve annotation of variants. We're thinking that this should really work closely with open targets and also probably with the knockout catalogue at the Broad. And here's just an overview of the GSIP proposal. It's in the safe haven. But essentially, we're talking about improving the classification of LOF using machine learning, validating the LOF using in silico and cellular models, cataloging the variants, and then improving understanding of the variants at population level. And just to summarize, what can LOF do for drug discovery? I think, hopefully you've seen it here. It can validate targets, the damaging variants. We've been doing that for a long time. The bypass of essentiality I'm really excited about, and could potentially identify alternative targets, could actually expand our understanding of what is druggable. Safer targets can be found with benign LOF, and all of the LOF data, I think, can tell us about target systems and interactions. And I will stop there. And we, as I say, we've got a growing GSIP project in this area, and we really would encourage others to join us in that project. We'd really be interested to collaborate. Thank you.